Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today in this video, I will discuss a very important practical. I will perform a very important practical that is related to bioinformatics. And the title of the practical is that how to perform blast on a protein. Okay. And then after that, I will tell you guys that how to understand the results. Okay. I have made a video over blast, over the introduction of blast. So please watch that video first. It will help you a lot. So then after performing this practical, you will then came to know that how blast is performed. So by watching that video first, you will have an idea that what am I doing in the blast. Okay. So that video is related to the introduction of the blast. So first watch that video first. Okay. So after that, then perform this practical. So then you will easily understand that how you will perform this practical. Okay. So let's start with the practical. Okay. I will perform the steps in a slower way. Slowly, I will perform the steps. So you should concentrate. Okay. First of all, the first step is you have to open the Google Chrome. Okay. And in the search box, you have to type NCBI. Okay. I have a recommendation. So if you are not having, so type NCBI. NCBI means National Center for Biotechnology and Information. Either you can click on this or you can directly click on blast. But I want to tell you that just click on the National Center for Biotechnology and Information. Okay. So from here you can click the blast. Okay. So before doing blast, you have to find out, you should have a amino acid sequence or a nucleotide sequence. Okay. It is very must. Okay. So I have made a video over in that how to find a nucleotide sequence or an amino acid sequence of a protein. But you have, but if you have not watched that video, so don't worry. I will also perform these steps in this video. Okay, because it is very much important for the blast. It is a major requirement for the blast that you should have a protein sequence. Okay, amino acid sequence or a nucleotide sequence. Okay. So first we have to find a amino acid sequence. Okay, we are doing amino acid blast. Okay, so first of all, what is the step? You have to open the NCBI database. Okay, and after that you have to click on the protein. Okay, if you are not having an amino acid sequence in the in your copy sorry, in your search box, so then you have to first find out the amino acid. Okay, so you have to click on the protein. After that you have to type a protein name, or if your examiner has given you a protein. Okay, so then you have to type its name. For example, I'm searching of reverse transcriptase. Okay, reverse transcriptase. So I have a recommendation. So I will take the human immunodeficiency virus 1 reverse transcriptase P51. Okay. So after that, you have to click on the reverse transcriptase P55. Okay. Or you have to directly click on the FASTA. It is much easier. Okay. You will skip a step. Okay. Okay. So here is an amino acid sequence. Okay. So you have to choose all the sequence. Okay, this sequence start from this proline. Okay, but you have to also select this first line. Okay, because this first line is very much important because it contains information related to this amino acid sequence, and it is necessary for the blast. Okay, so you have to click on the copy. After that, if you want to open a blast, okay, so it is also, you can open it, or you can directly click on the run blast. Okay, so then you can perform a blast. So if you are in a hurry, so you can directly click on the run blast, or if you are not in a hurry and you want to do it in a reliable way, So just open NCBI, okay, NCBI. After that, you have to click on next and center of this, and after that you have to click on the blast. Okay, so now you are doing a protein blast. Okay, you have taken an amino acid sequence, so you have to click on the protein blast. Okay, what is a protein blast? I have mentioned in the introduction of the blast. Okay, so there, first watch that video first. Okay, so you are having an amino acid sequence, so you have to click on the protein blast. If you are having a nucleotide sequence, so then you have to click on the nucleotide blast. Okay. What is blast X and T blast? And you have to not, we are not having any work with it. Okay. Because if you are a bachelor student, so these two blasts are important for you and enough for you. Okay. So click on the protein blast. After that, you have copied that amino acid sequence, so just paste that your amino acid sequence here. Okay, and no need to do other don't click on the other parameters, just leave it here. Okay, you are doing a protein blast. Okay, so it's okay. After that, you have to click on the blast. Okay, so it will take a minute or a two. Okay, it will take some time. Okay, so after some time, just wait a minute. Okay, the result have appeared. It have taken about 1 minute and 50 seconds or 2 minutes. Okay, so after that, this is the result. Okay, so what does this means? Okay, I have also shared the in the previous video that what does this means, but I will also tell you that what does this result means. Okay, so here is your result of that, I mean, reverse transcript test. Okay, so how will you identify that that my sequence is aligned with other sequences? Okay, we perform blast to align our sequence with other sequences. Okay, so and what is the main purpose of the blast? So I have mentioned in the previous video. Okay, so here is the result. Okay, so I want to show you a simple result. Okay, so if you click on the this gag ball human immunodeficiency virus. Okay, okay, so here is an alignment. Okay, I want to tell you guys that this alignment, this query alignment, is your alignment that we have entered in that select search box. Okay, and this subject is that the alignment of this protein. Okay, I want to show you. Wait a minute. Mm, okay, wait a minute. Okay, this one. Sorry. This cake ball human immunodeficiency virus. Okay, so this is this result. Okay, so our subject is, our query is our that we have entered the amino acid sequence in the search box, and our subject is the uh, this Gag-Paul immunodeficiency virus. So it has compared that sequence with our query sequence. Okay, and it is at the top. Okay, so this means that this the subject, the sequence of the subject has totally aligned with our query sequence. Okay, just look to the results. Here is our query sequence, amino acid sequence, and here is the subject. Wait a minute, I want to show you easily. Here is the query, okay, that we have entered in the search box, and here is the subject that we have clicked, that Gag-Paul immunodeficiency virus. Okay, a type of protein. Okay, and the middle line shows that how our sequence is aligned. Okay, so it is 100% aligned. Okay, just look at the identities. Okay, 440 by 440, it is 100% identical. Okay, so we can say that the sequence that we have taken, that we have entered in the search box, is totally identical to the subject that we have clicked on, that Gag-Paul immunodeficiency virus. Okay, so this protein is totally aligned with our query protein. So this means what is 1 to 60? So here is the these are 60 amino acid, okay. And the subject, the subject, okay, the amino acid sequence of the subject. So 588 to 647, these amino acids are totally identical to our query sequence. Our in our query sequence, there from 1 to 60 amino acids are identical, 100% identical to the 588 to 647 query subject sequence of that cake polyimmunovirus, okay. So that protein, okay. So this means that this 1 to 60 amino acids are totally identical to 588 to 647 amino acid sequence of the subject, okay. So this portion is totally identical to our query. Just look to this one, from 61 to 120, okay. We have taken again 60 amino acids, okay. I told you in the later video, previous video, that first the blast will make fragments of our query, okay. It will make fragments of our query. After that, it will compare it with different fragments, okay. So here is a sub fragment of a subject, okay, from 648 to 707. It is a 60 amino acids, okay. It is 60 amino acid, and we are also having, this is also 60 amino acid, okay. So from 61 to 120 amino acids are identical, are aligned with 648 to 707 amino acid of the subject, okay. तो इसका मतलब ये है, ठीक है, कि जो हमारे क्वेरी का ये वाला फ्रेगमेंट है, ये आइडेंटिकल है जो सब्जेक्ट वाले सीक्वेंस का, जो 648 से 707 तक है, ठीक है. तो इसका मतलब ये है, ठीक है. So this was all about the blast, that how you have to perform the blast, ठीक है. ये देखें, ये और हमारे साथ अगर मैं पीछे चला जाऊँ, ठीक है, तो ये डिफरेंट सीक्वेंसेस हैं, ठीक है. ये जो मैंने पहले आपको डिस्क्रिप्शन में दिखाया, ठीक है, तो ये तो सिर्फ मैंने गेक पोल ह्यूमन इम्यूनो डिफिशिएंसी वायरस ये अलाइन हुआ है. ये बाकी जो है ये भी अलाइन होते हो सकते हैं, ठीक है. और ये हुए हैं तो इसलिए इन लोगों ने जो है NCBI वालों ने रिकमेंडेशन में दी है. अगर आप इसको देख ले, ठीक है, तो जितना मैक्स स्कोर ज्यादा होगा, ठीक है, तो उतनी अलाइनमेंट ज्यादा होगी. देखिए टोटल स्कोर 920 जो हमारी क्यूरी कवर है, जो हमने जो क्वेश्चन दिया था, ठीक है, जो क्वेरी दी थी, तो उसकी 100% कवर हुई है, ठीक है. इट इज 100% कवर. एंड हियर जस्ट लुक टू द ई वैल्यू. ई वैल्यू मींस एरर वैल्यू, ठीक है. तो हियर देयर इज नो एरर इन आवर in this results, okay. If there is error, they will show you that 10 raised to power minus 14 or minus something, there is error, okay. It will show you percentage identity. Our first protein, जो gag pole protein, it is 100% identical, okay. Just look to the the second protein, so it is 99.77 identical, okay. So here is the I will just look to the accuracy, okay. So excision, this all are different parameters. If I want to show you another one, just taking a 98% identical, okay. Rec name, full gag pole polyprotein, okay. This is also a protein. It is aligned with our query sequence, okay. So just look to the alignment, okay. So it is aligned, okay. Just look to, so 439 amino acids are totally identical, and just one amino acid is not identical to our query sequence. So this is just because it is 98% identical, 99% identical, okay. There is no gaps, okay. You are not seeing any gaps, okay. So this there are no gaps, okay. So this was all about the blast, okay. There are many sequences that are not fully identical, or they are 50% identical. And example I have shown to you in the earlier video, so you can watch that video, okay. So I think now you are you are knowing the steps of that, how you will perform a blast. It will take about 2 minutes, or if your query sequence is larger, so then you can it will also take 3 minutes or 4 minutes, okay. With blast you can do many things. I will show you in the later practical, that what you will do with blast, how many things you can do with blast. Okay, so this was very basic, that if you are performing a blast, so how will you perform a blast, and when you the result is occur in this on the screen, so then how you will understand the results. Okay, that my query sequences how much identical, so these all are the results. Okay, so hope you have understand this video. If you have any question, you can ask me in the comment box. Okay, thank you very much. God bless you.